And thank you all for joining the APS seminar today on this beautiful afternoon. My name is Yuki, and I have the privilege of introducing Julija, who is our speaker for today. Julija Cubins grew up in Buffalo, New York, and went to high school and college in Vermont. She received her bachelor's degree in ecological agriculture from the University of Vermont in 2015, and got her master's degree in agronomy and plant genetics here at UMN in 2019. She had worked on a diversified vegetable farm in college, which spurred her interest in agriculture. She also became interested in research after working on an IPM hops project as integrated pest management, and she has also cultivated a love of working directly with farmers through working for Extension after her college experience. This combination of work made her realize that she wanted to pursue a graduate degree to learn more about agriculture and contribute to giving farmers more sustainable livelihoods. Julija is very interested in learning about the intersection between agricultural profitability and sustainability, and specifically working with crops that can contribute both to farm environmental stewardship and economic resilience. She enjoys working with a multidisciplinary team and is grateful for the opportunity to pursue research interests outside of agriculture. And so now she will give her talk, from pod to pod, bridging the gap between research and communication. So let's all give a warm welcome to Julija.
Great, thank you, Yuki, for that introduction. Thank you again for joining us today. It's a beautiful day to be outside, so thanks for taking some time to come listen to this seminar. My clicker does not appear to be working. Let's see. Neither does my slides. Okay, looks like it's going now. So to start, I want to give you an outline of what I'll be talking about today. I'm going to begin by talking about the shifting land use in Minnesota, and then transitioning into alternative crops that can meet environmental and economic demands that we are facing right now. This will bring me to my first research project, talking about corn relative maturity in Camelina, and then I'm going to talk briefly about another research project I have, Non-shatter Pennycress by Harvest Aids. That will bring me into a second introductory section looking at current methods and pitfalls of communicating scientific information. And then I'll talk about my third research project, Hooked on Science podcast. So again, the first thing I'll be talking about is shifting land use in Minnesota. As you're probably familiar, corn and soybean dominate the landscape in the upper Midwest and in Minnesota. In 2019 alone, there were 7.3 million acres of corn and 6.7 million acres of soybean grown in the state of Minnesota, which accounts for an enormous amount of the agricultural land that is cultivated. And so in a typical rotation, this is generally a 2-year rotation where 1 year we have a corn monocrop grown between April and October, and then we have a soybean monocrop that's cultivated between May and October. And what that means is we're using a very small part of the growing season, or we're using a very small part of the year to grow our crops. And this leaves us with about November through April where the land is fallow. And so what that means is that there are opportunities outside the corn-soybean rotation where we can grow crops. And there are also some externalities associated with this fallow period. And if you attended last week's seminar with Yuki, we covered a lot of this. And so I'm just going to briefly mention it. But when we aren't growing those crops, the ground is susceptible to erosion, to nutrient loss. And we're also not utilizing all of the solar opportunities that we have throughout the year. And so what that means is that these are underutilized opportunities where we could be producing a crop. And this is really important because our world population is growing. By 2020, this year, we should be reaching 7.8 billion people if we haven't reached that already this year. But by 2100, we'll reach about 10.9 billion people, and that population is expected to plateau then. But this is an enormous amount of people that we need to be able to feed high nutrient, caloric, calorie-dense foods to. We also need to be producing crops for fiber and crops for feed for animals and crops that can produce fuel. And so one way that we can do that is through temporal intensification, which is increasing the number of crops grown in a given period of time by using more of the growing season. And so that is really taking those underutilized opportunities that we currently have and placing crops onto them. And there are two ways that we can do this. The first is double cropping. And so if we consider our timeline of the corn-soybean rotation right now, we have that summer annual crop growing from April to November, and then fallow land between November and April. And what double cropping does is essentially just replace that fallow land with a secondary crop. It may place a little bit of constraint on that summer annual cropping system, either in the spring or in the fall, but overall, the summer annual crop is able to grow in its desired space where it's already been growing. The other option is relay cropping. And so relay cropping is similar to double cropping in that we are producing a secondary crop during that time when the land would otherwise be left fallow during our current corn-soybean rotation, but we have an overlap either in the fall or in the spring of that summer annual crop and the secondary crop. And if you look on the right-hand side of the screen, this picture is a representation of a relay cropping system where I relay cropped soybean into Camelina, and that gives you a little bit of an idea of what I'll be talking about later. However, there are some constraints with this system. First and foremost, we need to consider winter hardiness. In Minnesota, the minimum temperature ranges between -20 and -40 degrees Fahrenheit. And just where we are in St. Paul, that minimum temperature is about -20 to -25. And that's represented by this blue area on the map. But that's only about that is only about a third of the area of the state, and the other 2/3 get even colder, down to that -40 degrees Fahrenheit. So the crops that we're placing into that currently fallow period really need to be able to survive the winter. Another consideration is a short growing season. While there are about 6 months of the year where the land is currently left fallow, not all of those months a crop is going to be able to grow on. And so if we just consider this timeline again, we really need our crops to be able to withstand the cool temperatures going into November and December, and then the cool temperatures at the beginning of the year as well. And so what this really means is that we need our crops to accumulate growing degree days at low temperatures. And for those of you who aren't familiar, a growing degree day is a method to assign a heat value to each day. So each crop is assigned a value, and this is designated through experiments, what the lowest temperature it can begin growing at, and then usually a ceiling temperature that it won't grow if the temperature reaches above that. And so this is just kind of a graphical representation of that, where we have that lower threshold and that upper threshold, and the period in between those two thresholds is when the crop is accumulating these growing degree days. And this can be a way that farmers or researchers can look ahead to see when is a crop going to be mature, when is a crop going to be flowering, other important milestones like that. And so what we really need from these these secondary crops, is we need a lower threshold for that base temperature. Just for reference, corn and soybean have a base temperature of 10 degrees Celsius, which is about 50 degrees Fahrenheit. But we need something that can grow at much cooler temperatures than that. And then finally, we need crops that are economically viable. Farmers have a hard time putting crops onto their farms and onto the ground if those crops are not profitable, if they cannot at least break even the cost of putting them into the ground and producing them. But what is even better scenario for them is if those crops can make money above that point. And so this brings us to the second part of my talk, which is thinking about alternative crops to meet environmental and economic demands. And if you're at the University of Minnesota, you've probably heard of the Forever Green Initiative and their work on alternative crops. And this is really a multidisciplinary team that is approaching different crops to diversify land use. And so this is a suite of crops And what we're really considering is from start to finish, how can we get these crops on the landscape? So this includes geneticists, breeders, agronomists, food scientists, economists, and many other disciplines to really consider all of the steps that are required to get new crops on the ground and to make sure that they're profitable for farmers. And so that purpose is to develop this suite of alternative crops. There are about 13 of them at this point that are being considered. And what we really want is to give farmers opportunities to become more economically resilient and increase their vegetative cover, so we're not experiencing those externalities of losing soil, losing nutrients, and not utilizing all of our growing degree days. One crop that is a good fit is Camelina. So Camelina has been naturalized to many environments. It originated in Southeast Europe and Southwest Asia, but has been grown across the United States from the Pacific Northwest to the arid Southwest and to the Great Plains region that we are in here in Minnesota. It's also a winter hardy annual crop. So it's planted in the fall and harvested in the spring. And if we consider these pictures here, this was a picture taken in November 2019. Camelina was planted in September 2019. Just as a point of order, it's not fertilizer that's on the ground of this picture, it's actually pellets of snow. And we can see that this picture taken in the same place of this plot in April, the Camelina not only survived the winter, but also began growing again once it got warm enough. And Camelina also has a low base temperature for growing degree days. So it's been calculated that the base temperature for Camelina to grow is -0.7 degrees Celsius, so just below freezing, and that will allow it to accumulate those growing degree days into the fall and early winter, and then again very early in the spring when most crops would not even be able to be put on the landscape. And another important factor is a short life cycle. So as I mentioned earlier, those 6 months of the year that are fallow are not quite habitable for most crops. They're not going to be accumulating those growing degree days during a lot of that time. But Camelina has been shown to mature as early as 1,200 growing degree days. So on these figures on the right-hand side, on the x-axis, we have cumulative growing degree days, and on the y-axis is seed yield, and this is at 2 locations in Minnesota. But if we consider these points, these are different sampling points throughout the Camelina maturation, and any points that share an A or share any letter are shown to not be statistically different from each other. So any points that share an A are the maximum seed yield value, are not different from each other. But if we consider this first A value, we can see that Camelina matures as early as 1,200 growing degree days. And as far as seed content goes, this is a similar circumstance where that first period of maximized maturation is around 1,200 cumulative growing degree days. And if we just consider other winter annual crops, for example, winter canola is a very closely related species to Camelina, and that takes about 1,400 cumulative growing degree days to mature. So you can imagine that we can get Camelina off the ground more quickly than some of the other winter annual species in order to plant our summer annual crop. And one final note is economic viability. So Camelina is an oil seed crop, meaning that it's harvested for the seed, which is then pressed for oil. Camelina seed generally contains between 30 and 49% oil, with an average being about 40% oil content. And so that oil has 2 purposes. It can be used as an edible oil, which is advantageous because edible crops are sold at a higher premium than industrial crops, generally speaking. On the right-hand side of the screen is an example of Camelina oil that's produced in Quebec. It's high in omega-3 fatty acids, which is kind of a buzzword in food and nutrition, but it really is a heart-healthy oil, and that makes Camelina quite attractive as an edible oil. And similarly, I'm sure many of you have heard about the Impossible Burger and learned about this increase in interest in plant proteins. And so that's exactly something that we're also trying to maximize and look at with Camelina, since the plant protein market has increased quite a bit just over the last few years. Between 2018 and 2019, the value of plant-based foods went up 11%, and the year before that, they had gone up 15%. And if we compare that to the dollar value of the overall US food market going up, it's only gone up about 2% in both of those years. So these plant-based proteins are really making up a huge portion of the increase in the US food market. And this is something that we are considering right here at the University of Minnesota at the Plant Protein Innovation Center. Camelina, as well as other crops, are being considered if they could be the next ingredient in the Impossible Burger, which would be a huge selling point to grow Camelina. One caveat to this is that Camelina does not have GRAS status in the United States yet, and GRAS stands for Generally Recognized as Safe. It's an FDA term, and it really just says what it says in the title. It's generally recognized as safe. It can be used in food crops or food products per se, or I should say, without having that food crop, sorry, I keep saying food crop, food product need to be evaluated each time Camelina is added into something. So that is one barrier that I just wanted to mention to putting Camelina in food products in the US, though it is approved in both Canada and in countries in Europe as a food product or for use in food products. But in the meantime, Camelina oil can also be used as an industrial oil. And by that I mean it can be used as a feedstock for diesel or as a to make biodegradable plastics. However, this is not without challenges. We need to consider how we're growing Camelina before we can put it in any of these food products or any of these industrial products. And one of those challenges is that Camelina seed is very small. If you consider this picture on the screen, we have a penny for scale, and you can see that Camelina seed is very, very small. And so what that means is that Camelina does not establish well in a high residue environment. So if we are planting corn, harvesting corn, this is what a grain corn plot would look like after grain corn harvest. And you can see that there's all of these leaves that are left in the plot. And we consider this, it's called stover. And that makes up all of the above ground parts of the corn plant that remain in the field after grain harvest before it has been weathered or incorporated. And this residue provides a lot of challenges to getting Camelina into the ground. Because it is such a small seed, we need to make sure that it has good seed to soil contact, that it gets enough light, and this can be a real obstacle. And one solution is that we can remove corn stover from plots before Camelina planting in order to make it a better environment for Camelina germination and emergence. But this also comes with its own set of challenges, and stover removal can have negative consequences. In this picture on the right-hand side, you can see a plot where the stover has been removed and it's just bare ground, which is great for Camelina planting, but this means that the soil is more susceptible to erosion, meaning that if there's wind or rain, we can be losing that organic matter that's on the top of the soil, or the nutrients that farmers have applied, such as nitrogen or phosphorus. And removing the stover can even decrease the yield of crops that come later. In this series of studies done by these three authors, the first author looked at crops that were planted after stover was removed from the plots and found that there was a yield decline. And then the two subsequent authors came back and found that in that same area when they planted crops, there were yield declines in the plots where the stover had been removed 3 and 10 years later even. So this is also a concern. And then removing the stover can decrease the soil organic carbon. And soil organic carbon is important because removing it can lead to degraded soil structure, decreased aeration and water drainage, and can also decrease the nutrient holding capacity of the soil. It's not just affecting the carbon, it's also affecting all the other nutrients that are applied and are within the soil profile. And just to give a brief overview of the carbon cycle and kind of what's happening in this system, there's a lot of different pieces, and I'm gonna highlight them as I go through them. So when we have the corn harvest and that stover is on the ground, this is putting a wet organic material onto the soil surface. And this wet organic material, this stover, can be incorporated through tillage and then through microbes breaking down that carbon and adding it into the soil profile. And even when we don't have tillage, we can still have some of that incorporation through the microbial biomass. At the same time, we also have root incorporation where the plant is adding carbon to the soil profile. So both of these processes are adding carbon. Meanwhile, we have processes that are losing carbon from the soil, like root respiration and mineralization. And so as these processes occur, we are having carbon dioxide emit from the soil, essentially losing carbon. And this is important because even as we're adding carbon, or if we're not adding carbon, we will be losing carbon dioxide, carbon in the form of carbon dioxide, through root respiration and mineralization. So it's important that we're supplementing the carbon we have in the soil to ensure that we can maintain that soil organic carbon. And the good news is that it's hypothesized that winter crops may be able to supplement the soil organic carbon even when the stover is taken off the soil. There have been promising results in cover crops that they have reduced the impact of stover removal in both continuous corn and corn-soybean systems like what we're looking at here. However, this research hasn't actually been conducted, it's just hypothesized, so the actual research needs to be conducted to determine whether the winter oilseeds can provide the same benefits as the stover that would be left on the ground after corn harvest. And so this brings me to my first research project looking at corn relative maturity and Camelina. So I split up my objectives for this project into agronomic and environmental objectives, and I'm just going to focus on the agronomic objectives first. So my first objective was to assess the yield potential of a corn Camelina soybean rotation that utilized short season corn hybrids. And I hypothesized that the short season corn hybrids would yield less than the full season corn hybrids, that Camelina would establish more successfully following a short season hybrid than a full season hybrid due to the fall growing degree day availability. And soybeans really cropped into Camelina will develop fewer pods than soybeans that are planted into a fallow plot. And then finally, I thought that overall treatments with good Camelina establishment would yield the highest. And my second agronomic objective is to evaluate Camelina establishment in environments with and without corn stover. And I hypothesized that Camelina will more successfully establish in plots where the corn stover is removed. And for this experiment, it was run over the 2019-2020 growing season at 2 research sites in Morris and Rosemount, Minnesota. It was a randomized complete block design with 4 applications, and this study was funded through the Walton Family Foundation. And so in order to carry out this experiment, I used 3 corn hybrids, an 82-day corn, a 90-day corn, and a 95-day corn hybrid. And all of these corn hybrids were planted on May 7th, 2019 at both locations. And so nothing happened over the corn season. The corn grew and matured, and once it matured, we harvested each relative maturity at its when it reached its own maturity. So we didn't harvest these all at the same time. We harvested them as they became mature. That 82-day corn was done at about the last week of September. The 90-day corn matured around the end of the first week of October, and that 95-day corn matured around the 3rd week of October. And following harvest, we took seed yield measurements, took test weight on those those corn samples, and then that seed will be analyzed for carbon-nitrogen ratio. And after this occurred, we went back into those plots. This is a split plot design where within each corn treatment, we left half of the plot with stover remaining and then raked out half of the stover. So now we effectively have 8 treatments. And just for a visual representation at Rosemount, this is after the 82-day corn was harvested last September. On the left-hand side, you can see the plots where the stover remained. On the right-hand side, the plots where that stover was removed. So you can see that there is a very different planting environment for the Camelina depending on which plot we're planting it in. And then again at Morris, on the outsides of the screen, you can see the areas where the stover stayed on the plot. In the middle, the area where the stover was removed from the plot after that 82-day corn. So following corn harvest, either on that day or the next day, Camelina was planted into one of the 82-day corn plots, the 90-day corn and the 95-day corn, with both the stover removed and the stover remaining. And one 82-day corn plot was used as a control. So the plot that the stover remained on acted as a grain corn control, and the plot where the stover was removed acted as a silage corn control. And about 2 weeks after the final Camelina planting date, we went back out again and looked for establishment. So we did this in 2 ways. Populations were taken in 1 meter row of the plot, and canopio measurements were taken in a 1 meter by 0.76 meter area of the plot, and that's essentially just the width of the corn row by that same area that the populations were taken in. And so this was taken in the fall, 2 weeks after the final planting date, as I said, and then again in the spring after thaw, so we could see if the Camelina survived the winter and how it was growing. And for those of you who are not familiar with Canopio, it's an app that was developed at Oklahoma State University. And if you take a picture like this one on the left, where you have the Camelina, there's some snow on the ground, there's some corn stover remaining, it will take your image, translate the green pixels into white, all the other pixels into black, and that will give you a % ground cover measurement. And I was a little bit worried with the snow on the ground. It's pretty big. It was pretty pelletized when it was coming down that day. But as you can see, Canopio handled it really well, and we don't see much interference from that snow. It's really just picking up the Camelina in the plot. And just for reference, I won't be sharing data today, but this was our best Camelina plot in the fall, and it was planted on that earliest Camelina planting date after that earliest corn harvest with the stover removed, and it only had 1.7% ground cover, and we really saw a range between this plot where you can see that there's Camelina that established pretty well and Camelina that didn't establish quite so well in the fall. And so that Camelina began growing in the fall. We checked it again in the spring, and as it began growing, we planted soybean into it before the Camelina started bolting, which means that it was before the stem started to elongate and it got taller. And so at Morris, all of these soybeans were planted on the same day on May 7th, 2020. At Rosemount, we ended up having 2 planting dates, May 8th, and then again on May 20th. Our last Camelina planting date didn't emerge so well in the fall, so we wanted to give it a little bit more time to make sure that it would not all die when we planted soybean into it. And so following the soybean planting, Camelina was harvested. Camelina was harvested at maturity. However, if you look at the photo on the right, we were unable to combine at Rosemount because the soybeans were so tall in comparison to the Camelina. They were about the same height. And I would like to mention that this is pretty atypical. I had some conversations with some of my Forever Green colleagues who have been doing this for much longer than I have, and they concluded that this is a new thing that they have not seen. So this is really atypical and not something that we usually see, because obviously it would be a pretty big barrier to establishment. Farmers wouldn't want to establish a crop that they wouldn't be able to come in and combine. And this really speaks to our multidisciplinary team. If this was a huge problem, we would want to get Camelina off the landscape as early as possible. That is always the goal, but this would really mean that we need to step up that maturity. It needs to mature much earlier if this was a problem. And Camelina, as it stands now, you can consider these harvest dates July 6th. This is pretty late for a winter crop, considering that we want to plant a full season summer annual crop after that. And this speaks to our feedback loop with the breeding team, where they're trying to get Camelina lines that are going to mature earlier, so that if this case were ever to come up again, it would be less likely, because the Camelina would be at full maturity, it would be tall, while the soybeans were still short and really early in their growing cycle. And going back to these Camelina measurements, after the Camelina was harvested, hand harvested at Rosemount, as I already mentioned, it was combine harvested at Morris, taking seed yield, seed oil content, and then analyzing that seed for carbon nitrogen ratios. And after Camelina harvest, the only thing that's left is the soybean, and these are just beginning to dry down. They should be harvested by the end of October, hopefully. But before that happens, I'll go out and take counts on the number of pods and nodes in each plot, about 5 plants per plot, just to get an idea of how planting into Camelina has affected this soybean growth and development. But following soybean harvest, I'll take test weight, seed yield, seed oil content, 100 seed waste, and then analyze that seed for carbon nitrogen ratios. And this brings me to my environmental objectives. As I mentioned at the beginning, I had agronomic and environmental objectives. So this is the second set of objectives for this same project. So my first environmental objective is to assess nitrogen and phosphorus retention in the soil. And I hypothesized that the treatments with more ground cover will better retain their nutrients, and that really has to do with that fallow land being susceptible to erosion. If it rains or there's heavy wind, we may be losing nutrients. So treatments with ground cover should help to retain those nutrients in the soil. My second environmental objective is to observe soil carbon dioxide efflux throughout the growing season. And I hypothesized that carbon dioxide loss will be minimized in plots with greater amounts of living ground cover. So in order to carry out this set of objectives, I soil sampled 3 times throughout the season. I had a lot of help during this. You can see on the right-hand side of the screen, I have a whole host of helpers helping me with soil sampling. And this occurred after corn harvest, at spring thaw, and then again at Camelina harvest. And this was at 3 depths, 0 to 6, 6 to 12, and 12 to 24 inches. And these soils will be tested for nitrogen, phosphorus, and total carbon. And at the same time, I also took carbon dioxide efflux measurements. So this is really a measurement of the carbon dioxide coming off of the soil. And these were weekly measurements that occurred from April through Camelina harvest. And I actually did one in the week following Camelina harvest, when it would just be soybean. And this was only conducted in 3 plots in the 82-day corn. Looked at the grain corn control, where the stover remained and there was no Camelina, in the silage corn control, where the stover was removed and there was no Camelina, and then in the stover removed plot with Camelina planted, which really represents what we thought would be the best case scenario for Camelina to establish and grow in this cropping rotation. In order to do this, I used a Lycor 6400 portable photosynthesis system. You can see it on the right-hand side. And so what this does is, at the beginning of each measuring day, I would measure the ambient CO2 the ambient CO2 level, and set that on the machine. And I would then place this chamber on PVC collars. There were 2 per each plot. And it would pump the CO2 in the chamber down just below that ambient CO2 measurement that I had taken at the beginning of the day, and then measure how quickly that carbon dioxide built back up in the chamber. And this is giving us an idea of how much CO2 is coming out of the soil in each of these treatments. And so just to kind of wrap up this experiment, this field data collection will conclude with soybean harvest this fall. And it's an important caveat to note that carbon studies usually take multiple years, usually 5 or 10 years, to see meaningful differences, but this will give us a baseline idea of what is happening in a corn, camelina, soybean rotation. Because we are considering putting it on the soil, we need to know what's happening with it. And I would also like to mention again that this wouldn't be possible without the multidisciplinary effort across the Forever Green initiative with all our collaborators. And that kind of brings me into my next point, is that camelina isn't the only crop that can fit into this system. Another crop is pennycress, and pennycress is a good fit too for many of the same reasons that camelina is. And I'm just going to quickly go through this. It's similarly originated in Eurasia and is naturalized to many environments across Canada and the United States. Pennycress is a winter hardy annual crop planted in the fall, harvested in the spring. Camelina and pennycress are closely related species, so it makes sense that they share a lot of these same characteristics. It has a low base temperature for growing degree days. The pennycress base temperature is negative 2.5 degrees Celsius. If you'll remember from earlier, the camelina base temperature is negative 0.7 degrees Celsius. The pennycress can continue growing at even lower temperatures than camelina in the fall and then begin growing earlier, even in the spring. It also has a short life cycle, and this is really one of the reasons why we're interested in pennycress. It has a shorter life cycle than camelina. As I mentioned before, our camelina harvest date this year was July 6th, and that's pretty typical, just right around that 4th of July. But that's a little bit late for planting a summer annual crop either into it or after it. So pennycress matures about 2 to 3 weeks earlier, and there's been a lot of work done with the breeding team again to try and get that maturation even earlier. So we really like pennycress for that reason. And then finally, pennycress has similar economic viability. However, pennycress oil is mainly used for industrial purposes. At this point, we are still, not we, but as a team, the breeders and geneticists are looking at pennycress to create a line with an edible oil profile. And I believe that's in the early stages where at least one has been identified. But what we're using right now is industrial oil pennycress that could be used for, again, biodiesel or a biodegradable plastic production. And so this brings me into my second research project, which I'm going to go over pretty quickly. I just wanted to highlight that I'm working on a number of different things, and that is possible because as a group in Forever Green, we're all working on a number of different crops. There's a lot of crossover, and so we get to learn about a lot of different things and tackle a lot of challenges that are across many crops. And so this research project is Non-shatter Pennycress by Harvest Aid. And for this experiment, this was run over the 2019 and 2020 growing seasons in Rosemount, Minnesota. It's a randomized complete block design with 4 applications and was funded through the Minnesota Department of Agriculture. And so the big research question for this is, can applying a harvest aid to pennycress at physiological maturity advance maturation? And so a harvest aid is essentially either a chemical spray or a physical mechanism that's going to kill the plant as soon as you apply it. And so we're using this in order to kill the plant once it reaches physiological maturity, which in this case means that we have achieved maximum seed yield and maximum oil content. And can we apply this harvest aid and get that plant off the ground more quickly than if we just let it naturally senesce? And so I used a non-shatter line of pennycress. One of the challenges with pennycress is that the seed pods are very delicate and fragile as it nears maturation and can easily break open, and we call that shatter. But this is a non-shatter line of pennycress, so this shouldn't happen in this case. And it should still go through a thresher or a combine pretty easily when you use something as destructive as that mechanism, but not just a rain or windstorm would cause those pods to break open. And so I used 5 different treatments, 2 chemical treatments at 2 rates each, and a swathing treatment, which is essentially just a giant cutter bar that you can go through and cut down that pennycress with. And then there were controls where I took samples at both physiological maturity and harvest maturity. And for this data collection, I collected seed yield, seed oil content, biomass, and seed moisture at treatment application and at harvest, and then looked at seed shatter if we also want to see how well this non-shatter line does. And then following all of this data collection, I will do an economic analysis to determine if applying a harvest aid is economically productive for farmers or if it is not financially feasible for a farmer to do this. And so just some brief conclusions. As I mentioned, I was just going to briefly talk about this project. I did want to highlight that I had another field project. The field data collection finished this year for this project, and this research wouldn't be possible without our multidisciplinary team. And this pennycress really just represents one potential scenario to get pennycress on the landscape, and that's through using harvest aids. But there is a host of research being done on pennycress and camelina, as well as the other forever green crops, to try and get new crops on the landscape during that fallow period of the corn-soybean rotation. And so this brings me to a second introductory section of new materials, where I want to talk about the current methods and pitfalls of communicating scientific information. So as scientists, we typically communicate our science through scientific papers, through conference presentations, and in extension, we are blessed to have an opportunity to go straight to our stakeholders, to the farmers. And this is not the case in many scenarios, but these current methods of scientific communication are good at communicating our science to a fairly narrow audience. And we're really only reaching people that are interested in the same topics, whether it's scientists or farmers. And we don't really reach a general audience or even scientists in other disciplines. And as we come to a time where we can't have field days where we would normally share extension information and research information with farmers, and we are coming to a precipice where we can really combine that challenge with the challenge of communicating to a general audience. And so as scientists need to reach broader audiences, it's not just times like COVID that are spurring this. One other reason is that dedicated, nonpartisan science journalism is becoming obsolete. So there was a time when there were scientists in newsrooms who were also trained as journalists who could take complex scientific information and distill it into a way that a general audience could understand, really simplifying those concepts, but getting the core concepts across. But this is really becoming less and less frequent and just a less common occurrence in general. And so this means that there's rising expectations for researchers to communicate their research directly to the public, but we're not very good at this. And one of those barriers is because of the structure of academia. As researchers at often universities, the tenure track rewards publications and conference presentations, but it's not looking and targeting as much of that general science and dissemination to a more general audience. And there's some pitfalls of the way we communicate science when we do try to communicate to a general audience. One of those is we look at data subjectively, whereas that's a training thing. We, as humans, would usually look at something from an emotional standpoint. And our presentation styles are often at odds with how we would communicate to a general audience. And then finally, our outreach efforts need to be empirically supported. So ironically, we are not using the tools, generally speaking, that social scientists can give to us and show us how to connect to our target audiences. So we really need to be mindful of if the outreach efforts that we're doing are actually going to reach the people that we're trying to reach. So how can we balance that research with learning? One way that has been emerging and has been evolving is online programming, which is a great way to reach broad audiences without having to do a ton of legwork. And one tool that can be used for that is podcasts, which can be used as a tool to produce on-demand media. And for those of you who are not familiar, podcasting was developed in 2004 by a gentleman who was frustrated with having to download MP3 files manually and wanted an automatic way to do this. And the pod of podcasting comes from Apple's iPod, though it's not related to Apple in any way, and casting comes from broadcasting. And podcasting is not only a great tool to produce on-demand media, it's also a cheap tool to produce media. So anyone that has a smartphone can produce a podcast. There are plenty of free apps that you can record, edit, and upload your podcast in, and then it can be distributed. You don't really have to do anything in order to get that information to a broad audience. Whether it's actually getting to people is a whole different story, but it's really easy to create content that can be available to a wide audience. And one of the reasons that podcasts are attractive is that they can be played and replayed at the listener's convenience. And there's been growing research to suggest that many people are listening to podcasts at home when they're doing other tasks, whether it be folding laundry or washing dishes, as well as commuting. So podcasts are really a useful tool to reach people in time when they need some entertainment or they want to learn about something. And thankfully, podcasts has been used as a tool for science education. So as an educational tool, there have been quite a few studies looking at podcasting as a tool to review educational materials for undergraduate courses, as opposed to lecture notes or textbooks. Students are saying that they like podcasting as a way to review materials and relearn things that they have already talked about, concepts they already know. Podcasting has also been used as a way to reach general audiences by scientists looking at research, which is also promising. And then one recent study showed that education was a preferred podcasting genre. About 40% of the people surveyed in the study, there were about 1,000 participants. Those 40% of people indicated that they enjoyed using podcasting as a way to learn about new things. However, one pitfall of this, which should not dissuade anyone from podcasting, but it is important to keep in mind that popular science efforts can make a general audience disregard expert opinions, since we're taking these complex concepts and distilling them into something that's a little more simple while retaining that core information. It can cause a general audience to think they know more about the topic, more about that particular issue than they actually do, because those popular science articles or podcasts are generally being distilled from a wide variety of information and research studies. But again, not something that should dissuade anyone from podcasting about their research. And so who listens to podcasts? One recent survey, this has been the largest survey of podcast listeners, looked at about 2,000 participants who listen to podcasts regularly. Most podcast listeners fell in the age bracket between 25 and 45. So it's a decently young audience. And about 51% of those respondents had a college degree. However, this did not qualify the types of podcasts that people were listening to, just that people were listening to podcasts and more of the demographic aspect of this. And furthermore, we don't know why people are listening to educational podcasts. So to kind of put all that information together, we know people are listening to podcasts for educational purposes, And we know that a lot of people are listening to podcasts, but it's really important to understand why they're listening so we can better tailor that information to people that want to listen to educational podcasts. So as I mentioned earlier, podcasting has been used as a tool in higher education as a review tool before exams, and that's what a lot of the empirical studies have considered as far as podcasting as an educational tool. Another study looked at attitudes before a subject, or towards a subject before and after listening to a podcast. And then another looked at audience members, or a podcast that was directed at a specific audience. And so what I'm really interested in is who is listening to podcasts and why are they listening to podcasts meant for a more general audience. And so this brings me to my research project, Hooked on Science, my final research project that I'll be talking about today. And so my first objective was to create a podcast that would increase access to scientific research. And I hypothesized that podcasting would be a convenient way for people to learn about new topics, and that listening to a podcast would be a less intimidating way for a general audience to learn about scientific research. My second objective was to understand the demographics of those research podcast listeners. And based on those survey results that have already come out, I thought the majority of listeners would fall in that 25 to 45-year-old range. And because of the nature of the research-based podcast, I also thought the majority of listeners would have a college degree. And my third objective is to better understand why listeners are using podcasts as an educational medium, why this is an attractive way for them to learn. And so I hypothesized that listeners who want to stay actively informed will be listeners of this podcast, and also people that just want to learn more about a field of interest that they may not have much exposure to in their regular day-to-day life. And so there were three phases to this project. The first was creating and hosting a podcast. The second was collecting survey data from listeners, and the third is to hold follow-up interviews. And I would like to note that this project isn't funded by any specific group, but my time is funded by the Walton Family Foundation through that other project that I was talking about, the Corn Relative Maturity by Camelina. And so in this first phase of creating and hosting a podcast, the first step was to generate my own content. And this was by no means a requirement to study podcasting, but I was really interested in the process and just to learn more about how to conduct a research-based interview that's hopefully kind of intriguing and can get a lot of information across in a simplified way, but also in a factually accurate way. Guests are all recruited through word of mouth. I have had a number of guests in the Applied Plant Sciences program, especially at the beginning, but I've also recruited people through friends of friends, through Facebook groups, really anywhere that I could post about the podcast, and anyone that has expressed interest, I follow up with them and try to get them on the show. And before we actually get to recording, guests provide materials for me to develop questions. Usually this is an abstract or a poster, something that can give me a pretty well-rounded view of a project that they're working on, and so that I can make some general questions so that we can understand kind of the groundwork before we get to the actual research. Interviews are recorded remotely using Zencastr, which is an online podcasting platform. And then those episodes are edited using DaVinci Resolve 16. I upload my podcast to Anchor for distribution. There are many distribution sites, a lot of them are free, that take your podcast and then distribute it to places like Apple Podcasts, Spotify, any of the other podcasting platforms that you may listen to a podcast on. And then I manage social media accounts to promote episodes, promote any related information about guests who have been on the show, post any information about science communication that I see online that I think might be of interest to my social media followers, and just general content about science. And the next phase of this project is to collect survey data. And so the first part of survey data collection is quantitative data. And this will consist of demographic questions that are multiple choice, and then survey questions that utilize a 5-point Likert scale. And a Likert scale is, if you've ever seen a scale that goes from strongly agree, neutral, disagree, strongly disagree. That's a Likert scale. There is a lot of precedent for this, which was something that was important to me. This is data that I don't have a lot of experience looking at and understanding, interpreting. So there are ways to look at your questions themselves and validate them, and then to actually do statistics on the answers to those questions. And as a side note, I don't have that many listeners at this point. My listenership is steadily increasing over the course of the months that I've done the podcast. And so while I'm very excited about that, I need a larger sample size for survey data. So I'm going to collaborate with other podcasts that have similar missions to talk about scientific research in order to deploy my survey. And this is in development, these survey questions. I'm working on them, and they're set to be deployed using Qualtrics in early 2021. And then following that initial survey data collection, I also want to collect some qualitative data. And these questions will be developed after some of that quantitative data has been collected. And this will consist of face-to-face interviews over Zoom so I can better gain insight into the findings of the initial survey. And obviously participation is voluntary. At the end of the initial survey that uses the Likert scale, there will be a form that people can fill out if they are interested in having a follow-up interview, so I can talk a little bit more about with them about their podcast listening preferences. And I hope to conduct these interviews in early spring 2021. And I would like to thank all my plant sciences participants. As I mentioned before, I've had a number of people join me from the Agronomy and Plant Genetics department and the Horticulture department. And so I just want to give a shout out to them and thanking them, especially the ones who were early on in the process when I had, I was still figuring out what I was doing. I was still trying to figure out what producing a podcast looked like. So people that really went out on a limb and helped me out and had a conversation with me. And I'd also like to plug that if you or someone you know would be a good fit to participate on the podcast, please send me an email. I manage an email just specifically for the podcast. It's hookedonsciencepod@gmail.com, just to kind of make sure I don't get things lost in my school email. And I would also like to mention that this is not just a plant sciences podcast. This really is a general science podcast. I have had quite a few plant sciences participants, but we have talked about other topics such as theology. We talked about how video games shape views of national identity. We talked about anticoagulant rodenticides. So we're talking about a lot of different things. So I don't want to discourage anyone from participating, but if you have someone or you yourself would like to participate, please let me know. And to conclude, this is a constantly evolving project. I am currently in the position of making up those survey questions, so I'm still learning about this. This is in the very early phases of the science part of this, but it's evolving and I'm learning. And one other thing I'd like to note is that I understand that scientific research does not appeal to everyone. So I know not everyone's going to want to listen to a podcast about scientific research, but I hope some people will express an interest in that. And that this podcast can really provide some baseline information for research-based podcasts in the future. And I hope that podcasting can serve as a model for a new way to adapt our research for a general audience. And with that, I would like to thank all of you for coming and listening today, as well as my co-advisors and my committee members, as well as everyone who's helped me out on these projects through the past few years. And with that, I will take any questions.
Thank you, Julia. This is Don. I just want to let everyone know that we had decided ahead of time, and I gave permission for her to take the full hour because I thought it was a unique opportunity for her to lay out her full body of research and to show the diversity of those things that she was working on. So just so you all know, I gave her permission to lay it all out today for the full hour. And it's open for questions.
So if you have questions, there is a chat box. You can enter them in. And I think if anyone wants to ask me a question directly, you can raise your hand and we can get your video and audio so that you can share directly.
Looks like Axel has a question. I think we're going to open his mic up here. Axel.
Thanks. Yeah, thanks, Cody. Hey, Julia, thank you for your presentation. Very nice, actually. And I just wanted to first congratulate you. It seems like you just nailed it down. I see what you just presented as probably the future of extension, if I can say that. But anyways, I know that podcast is a good stuff. My son is a 20 years old guy, University of Minnesota student, and he is always podcasting. So you are right. So congratulations. And the question I have for you is, it looks like, the people who have helped you has increased from to more than 100, how to say, listeners in the graph you showed. How long do you think it will take for you to reach a higher number of people listen at your podcasts? And the second question I have is, how far are you planning to go with this idea during your grad studies?
Yeah, so I think I'll answer your second question first. I haven't really set a end date for this. I think I'll do it as long as it's feasible. I would like to do it throughout the remainder of my graduate school, my time in graduate school at least. I think it's been a great way for me to learn about other topics as well as a way for other people to learn about other topics. It's been really exciting to have some exposure to other research that's happening both in the plant sciences and outside of the plant sciences. So I don't have a particular end date. At the very least, I will have the podcast go on through the end of my survey data collection. I think that would be, the first maybe milestone that I would go back and reconsider if, there's time I need to allocate to other projects or my writing or statistics or what have it as I come to the end of my graduate school. And as far as your first question, how long it takes to get listeners, I think that is the formula that everyone's trying to crack. It's difficult because each podcast is different. Some people are looking for something more entertaining. And one of the things that I have tried to leverage is that I have a new guest at every episode and hope that they will share that and have like an agreement in my Google form that they fill out before they participate, that they will share that among their networks and post on, their social media pages. So mine is really through word of mouth. Certainly people can, pay to promote their podcast. But I've found that word of mouth has been somewhat successful. I post on different Facebook groups and look for guests in other disciplines, and that's mainly how I get them. So I think there's a lot of ways to go about it, and I don't know if I've found the perfect way for me to promote my podcast, but I'm trying a bunch of different things to just see what works. And I've seen spikes for certain episodes that it seems like people are really interested in learning about topics, not just in the plant sciences, but in other disciplines as well. So I'm trying to offer a variety to get as much of a general audience interested in my podcast as possible.
Cool. And may I ask you another quick question?
Sure.
Yeah, and are you guys looking at somehow to integrate this into the University of Minnesota Extension?
So there are a couple podcasts that already exist through Extension, but I haven't looked at that specifically, though it would be easy enough to repurpose that information, especially the topics that are relevant to, our plant sciences, to, then have kind of a separate feed that might be just Extension specific.
Thank you. Very good. Congratulations.
Thank you.
Julia, I think there's a question in the chat, but I had a question first about your survey methodology. I might have missed it, but you said you're going to be using other podcasts. So are those being are you going to take random samples from those other podcasts to take your survey?
Yeah, so it might not have been clear about this in my talk, but what I'm planning to do is, announce it on both my podcast and social media, and then these podcasts that I'm collaborating with, invite them to also share it with their networks. Hopefully for a couple episodes worth at least. I think I'll probably have my survey open for about 6 weeks. The listeners who fill it out early might get a little bit annoyed that I keep announcing it, but I'm really just going to have those other podcasts distribute it at their kind of convenience and how they're able to. So it will kind of vary from podcast to podcast, but I'm not going to specifically target, certain people listeners within each podcast. It'll be, who can I get to fill this out? Because I think numbers are going to be my difficulty, which is one of the reasons why I am, opening it up to other podcasts. I want to make sure I have a sample size that actually can show some, statistical information and give us some actually good, solid information about educational podcasting.
Great. Thank you for that answer. There is a question in the chat. I believe it's from Mary. Are you planning some podcasts with social science researchers?
Yes. So this is another thing as, a plant scientist, someone that's been in the plant sciences for a long time. I don't have as many connections with social scientists, but that is certainly included in the people that I hope to integrate into my podcast over the next few months, and some of the topics that I am interested in, exploring and talking about.
Great. And you said that anyone can be recommended for your podcast, correct?
Yes, correct.
So if people can just go to the website or email you and potentially get involved.
Yeah, that email again is hookedonsciencepod@gmail.com. And if you just send me an email on that, I have an infographic that kind of lays out the process. And I have a Google form I have people fill out, so I can get some information. I can learn what social media people are comfortable being tagged in, things like that, and then we can kind of connect and go from there.
Cody, Roger Becker raised his hand.
There you go, Roger. You can go ahead and ask your question to Julia.
Julia, I'm just curious on your podcast, how do people, sorry, I'm outside, the dogs are barking. On the podcast, how do people find you? Are there keywords or platforms or ways to be found in the sea of information that's being generated out there?
Yeah, so I think that's also one of the tricky things along with generating listeners is making sure you have a way for people to find you. So I have Facebook, Twitter, and Instagram feeds that can be found just through searching Hooked on Science. The podcast should show up. As far as a Google search, I don't know if I've actually Google searched my own podcast before, though my search results might be a little bit skewed, but I might have to try that in incognito mode. As far as, putting podcasts up online, they don't have you put keywords generally outside of that it's an educational podcast. So it could be, categorized on the different podcast apps. But that is an interesting question to consider.
Yeah, thank you.
Julia, how easy would it be? I've been thinking about some of these things for a long time in terms of all the good words that get spoken in this seminar series. And I was thinking a number of years ago, and just to tell you, the graduate students didn't buy into it, in putting forward something out for general consumption. What would it look like? Each semester we have graduate students making these presentations, and what has always bothered me that all of the things that students are working on really doesn't get out to the general public. So I was always thinking that this platform, the seminar series, could be a great place to start to teach those skill sets that you're talking about, to get this information out to the general public. Do you think this is a platform that could work off of?
So from what I've seen, I think something like this seminar, I know that these are recorded now that we're on Zoom, they're recorded, and I think they could be easily repurposed as YouTube videos, especially. A lot of podcast listeners, actually, one of the preferred sites is listening to a podcast on YouTube, which seems counterintuitive because YouTube is a video platform. But a lot of people like that, and I think especially in the seminar context where the visual aids are really important, it would be good that would be easily translated to YouTube. As far as podcasting itself, it might be kind of tricky because, as everyone who attended today, that was almost an hour monologue essentially, which may not hold listeners' attention for, that long. I think podcast is really great as an interactive, medium between the host and the guests, or the host and the host, however it's set up. But I think that this information, outside of the seminar itself, could easily be repurposed. Once we give our seminars, we have all this background information, all this research information that could easily have like a secondary sit down and have more of a conversation that was, structured based on, the seminar.
Yeah, what I had proposed to the students a number of years ago, and I had proposed it for several years in a row, was to actually set it up so they would take and present after they made their presentations, to do a 4-minute video. But the students showed no interest in it at that time. Maybe it's a different time now. Maybe we ought to go back and revisit that.
Yeah, and I think podcasting is also a good medium for slightly longer, things that require a little bit more information to build around and then kind of dive in. So a lot of podcasts are between 30 minutes and an hour. Most listeners prefer a podcast it's either like 30 minutes or 45 or 60 minutes. So there's a lot of space to, talk about a lot of these concepts.
Yeah. So are there any other comments or questions out there in the audience for Julia? If not, thank you for your attendance today, and look forward to seeing you all next Monday.